Okay, so if you've written down your responses here, then this is probably going to go very easily. What would be a positive result in this experiment? Well, again, we're expecting if there's microbes in the soil, they're going to oxidize that carbon, release carbon dioxide, and it'll be radioactive. So therefore, a positive result, which would imply there are microbes in the soil, that's what this symbol means, implies microbes in the soil, means that we would detect radioactivity in the headspace of this experiment. If there weren't microbes, what would happen? Well, if there weren't microbes, the sugar, would stay dissolved in the water and stick down here into the soil sample. And therefore, nothing would happen. The radioactivity would remain in the soil and it would not be detected in the air. Radioactivity isn't an atmospheric phenomenon. It's not something that can sort of float around in the air unless the thing that's emitting the radioactive particles is actually in the air. So the thing that's emitting the radioactive particles are down here in the soil, and they're emitting radioactivity out this way, but this isn't going to sample them because there's none of it in the headspace. So that would mean a negative result would mean no radioactivity in the headspace. Okay, perfect. That's the basic experiment. Now, of course, there's a ton of engineering, and I can't get over this. You know, there's a ton of engineering here. It's not like this is a very, very simple thing to actually build, but it's a simple thing to design. Because like I said, simple experiments are the way to go. So before we do this, though, before we stack this on top of a big booster and send it off to Mars in a multi-million, even now today, billion-dollar project, we better be sure that this thing works. Because if it doesn't, we're just wasting our time. So how do we do that? How do we double check? I mean, we're not dumb. We're going to make sure that this thing is going to work before we send it off. Okay, so how do we do it? Well, best thing to do is test it on Earth. And to do that, we therefore need to design experiments on Earth to be done by essentially the same thing as what's going to be sent to Mars, and double check and make sure it's working. That concept, doing an experiment under a condition that you know what the result is supposed to be, is what's called a control. Okay? The control is testing whether or not the experiment works. When we use the word control, that's what we mean. There's a common misconception. People tend to think that a control is simply something to compare something to. No, it's true. We are going to compare our result to the control result, but before we even look at the actual test result, we have to make sure that the test is working. We have to make sure the experiment's working. So, in this particular case, we need to run 2 controls and 1 experiment. The 2 controls need to be what we call positive and negative controls. So whenever we do an experiment, we always have to make sure that we have to do them to make sure that they're controlled. And in this case, our experimental control is just testing the validity of the experiment. Now, how are we going to do that? Like I said, we need 2. We need positive and negative controls. The positive control is testing the experiment under conditions where we know it should give a positive result. So here, a control means you know what the result is supposed to be. So we're going to test it under conditions where we expect or we are sure we should get a positive result. And if we do get a positive result, then there's no reason to believe the test is not working. If we don't get a positive result, then we're sure that the test is flawed. All right, and then that's only one thing, because it's possible we give a positive result every single time, whether there's microbes there or not. So we have to do now the negative control. We have to test it under conditions where we know there are no microbes. It should give us a negative result, will give us a negative result. If we test it and it gives us a negative result under those conditions, beautiful, we're fine. If we test it and it gives us a positive result, then we are certain it's flawed. Right? Now, this is a very common concept. We use it all the time in this type of experiment. Not every experiment needs positive and negative controls. Don't get that idea. Every experiment needs a control, but not every experiment needs positive and negative controls. Depends on the design. Here's another example of one that needs positive and negative controls, a test for HIV. How do we test a patient for HIV? Well, here's what we don't do. We don't take blood from the patient, run the test, and if it comes back positive, we say they're positive. If it comes back negative, we say they're negative. That's far too dangerous. Now, we might do that in the clinic, but we do a definitive test much more carefully. The definitive test is this. When we test a patient for HIV, we take blood from actually 3 people. We take blood from the patient who we're testing, we take blood from a person who we are certain does not have HIV, and we take blood from a person who we are certain does have HIV. We test all three of those people. Now, the person who we know has HIV is our positive control. We know that that test has to come out positive. The person who we're testing who's not HIV positive, who doesn't have HIV, we know is our negative control because we know that person must come out negative. The patient we don't know. The patient is the test. So, positive control, person who has HIV. Negative control, person who does not have HIV. Test the patient. Same exact logic here. Right? Now, what would happen? Let's think about this HIV test. What would you conclude if the positive person tested positive, the negative person tested positive, and the patient tested positive? In that situation, do not go to your patient and tell them that they tested positive for HIV. You don't know why they tested positive because the negative control tested positive. In other words, you tested somebody who is HIV negative, who does not have it, and they tested positive. Therefore, there's something wrong with the test. You don't know what's going on, so you don't know why your patient tested positive. Okay? What result would allow you ethically to walk into a clinic and tell the patient, yes, you tested positive? That would be a test where the positive control came out positive, the negative control came out negative, and the patient came out positive. In fact, it's even more complicated than that. When we do this for real, we actually do 3 tests. All of those 3 individuals are tested 3 times, and then oftentimes it's done even in a separate lab. So there's a total of 6 tests on those 3 individuals. And they all have to come out exactly that way. Positive control positive, negative, patient positive, for us to walk into the clinic and tell the patient that they tested positive. Now notice when we do that, we don't say, you have HIV. We don't say that. Although well, some people do, but we shouldn't. What we say is, you tested positive, because even with that, there's still a possibility that the test is wrong. Now this leads back to this problem on Mars. We're going to do an experiment. We're going to do this test, and we're going to test it before we send it up there. Even if the experiment passes these control tests, there still might be a flaw. There still might be something wrong with it. We are not trying, therefore, to prove anything. I do not want you to think of science as proving things or disproving things. We don't do that. We can't do that. Proof requires perfect knowledge. We don't have perfect knowledge. The question that we're always dealing with, and this is true in the laboratory and true in all the rest of your scientific endeavors, the question we are always dealing with is simply this. Do these data support a particular conclusion? Do they contradict a particular conclusion? Or do they say nothing about a particular conclusion? That's the only thing we're trying to determine. So from now on, whenever you write or speak and you talk about scientific proof, I want you to stop and think, do I really mean proof? Do I really have perfect knowledge here? And I'm going to test you on that. Because this is one of the most critical issues, one of the most critical disconnects between the scientific community and the community at large. The community at large is always talking about how scientists prove things and disprove things, and that's not what we do. We never talk about that amongst ourselves that way. So this is something then we've got to get out of our mind. If you want proof, you got to go to bad philosophy or mathematics. In mathematics, you can have perfect knowledge because everything you're talking about is abstract. But not in science. We can't do that. In science, all we can do the data support, contradict, or say nothing about a given conclusion. Okay, so keep that in mind as we move forward because we're going to immediately apply that concept. Okay, so let's do this. How would we do a positive control for this experiment? How would we do a positive control? How could we run an experiment that is guaranteed to give us a positive result? Well, do it on Earth where we know there's microbes. In fact, let's do it in a place where we know there's lots of microbes in the soil. And that's exactly what they did. And I believe that this picture here of Carl here sitting here with this mock-up of the lander was actually in the place where they were doing that test. This looks much like Mars. This is part of California. And it was in an area, though, where even though it's in California, it doesn't look like there's anything alive there. There's actually quite a bit of microbes in these soils. So that's our positive control for this experiment. Run the experiment where we know that there are microbes, and that was done in California. But then how do you do the negative control? Well, you'd have to do it where you know there are no microbes. Well, where on earth are we going to go where there's no microbes? That's a problem. Everywhere we've looked, even in Antarctica, even in places in Antarctica where it is so cold it can't snow, they're dry valleys, In fact, it's so cold that CO2 will freeze. We find bacteria, we find prokaryotes that are living there. So that's a problem. But here's what we can do. We can kill the microbes that we know were there. How do you do it? Heat them up. Same thing you do when you cook. Same thing we do when we do surgery. We heat the living daylights out of something, and that kills all the microbes. All right, so the negative control then is going to be this. We're going to take the exact same soil that we use for the positive control, but then we're going to heat it. And when we heat it up to the point where all the microbes are killed, and then we test to see if we get the negative result. All right, so let's take a look at that. There's our positive negative control concepts. Let's actually look at the download data from the lander. Here is the actual data from the controls. This was done in California by a mock-up of the spacecraft that didn't fly to Mars, but it was identical to the ones that did. On this axis here, you'll notice it says radioactivity and CPM. That stands for counts per minute. This is what a Geiger counter is counting these radioactive decays that hit its detector. And then on this axis, we have time, and it's noticed elapsed time in sols. Sol is a local planetary day. On Earth, a sol is a day. It's 24 hours. On Mars, a sol is slightly different. It doesn't rotate at the same rate Earth does. It's pretty close, actually, remarkably close, but it's not exactly the same. So we're looking at days here on Earth. Okay, now here's what they did. They went through this experiment right here. Remember what they did? They opened it up, scoop the soil, put it in there, close this, blow out all the atmosphere that was in there, replace it with helium, dribble that labeled nutrient onto the sample, and then we know, this is the control, that there are organisms that are going to oxidize that carbon. They're going to basically eat it, and they go through this reaction here. If I can get to it, this reaction here, and they are going to produce CO2. So we know that. We know that's going to happen. We've radio labeled that carbon, so we know then that the CO2 that's given off must be radioactive, and we're going to see if we can detect it. Okay? That's what we have here. That's what this curve is right here. Right? So over time, what do we see? Well, here's the punchline of this. Over time, the radioactivity measured in the headspace increases. That's what this curve says. It's nothing more, nothing less than that. That's what it's doing. Now, these little things right here are recalibration purges. They recalibrate the instrument there, so don't worry about that. They purge here. They open it up and blow out all the radioactivity in the headspace. They also did a second injection right there. In fact, the second injection means that, so they first put the radioactive labeled sugar water here, and the second injection, they did it a second time on the exact same soil sample without changing anything else, and that's what this is right here. And you notice it inflects up. It goes up a little bit more, a little bit faster, which makes sense. If there's microbes in the soil, they're eating sugar, you give them food again, they're going to crank out more CO2. All right, so that's the positive control. And the negative control then was this. They took the exact same sample. They didn't do anything else. They didn't open up the thing. They didn't put a new scoop in. They just took the exact same sample, and they heated it. They heated it to about 150, 160 degrees Celsius, and that's well above boiling. That kills all known microorganisms on Earth, so no problem. And then they repeat the experiment. They dribble more sugar water onto there, and this is what they get, in this case down here. Okay? Now, let me ask you this. Here's the data. You all have brains. Tell me, are we ready to fly to Mars? Okay, now, notice what I'm not asking. What I'm not asking is, does this prove the experiment works? Remember, we can't prove anything. There's no proof involved here. What we're asking is there any evidence from this data that the experiment is flawed? What do you think? I'll tell you what a professional thinks. The professional looks at this and says, yep, that's exactly what we would have predicted if the experiment's working. So here's the point, and this is kind of scary for a lot of people. When you look at this, the proper conclusion is there is no evidence that the experiment is not working. The evidence suggests the experiment is working fine. Now, that's different than saying the experiment is perfect. That's different than saying the experiment works. We're not saying that. We can't really say that. We certainly can't say it's perfect because we know it's flawed. Everything's flawed. But the point is, there's no evidence from these controls that the experiment is flawed. It seems to be working. Now, they tested it, of course, much, much more extensively than just this one test. But what I'm showing you is the conclusion from this test. They tested it a number of other ways. It passed all those tests. So they put it on top of the stack, shot it off to Mars. Did it a second time. Now here, are the actual data from Viking Lander 1. This is now what occurred on Mars. We sent the thing up to Mars, it landed, and we did these experiments on Mars. Okay, so now, same thing. Here are local Martian days, sols. Here is the radioactivity that was measured in the headspace. All right, so on Mars, we do this. Open, scoop, close, purge with helium, dribble the sugar water on there, and we got this, cycle 1. That's the data. The punchline is quite clear, and you should be able to pick this off now. Radioactivity in the headspace increased over time on Mars. Okay, so then they said, all right, now we're going to do this. Take that same Martian soil, heat it up to, again, about 160 degrees Celsius or so. There it is, 160 degree control. And dribble more sugar water on it, which they did, and then they measured this. Okay, so that's what they get after they heated it up to 160 degrees. Now, they saw that. We're going to stop right there and ask, what do you conclude? And again, we're not concluding anything about proof. We're not saying anything whether or not there is or there is not life on Mars. The question is this: are these results consistent with there being life on Mars? Are these results inconsistent with there being life on Mars? Or do these results have absolutely nothing to say about there being life on Mars? Right? Well, it's a little debatable. We can debate it. But this is certainly consistent. The results are certainly consistent with this conclusion. Something broke the sugar down and made it into a gas that was released into the headspace. Because the gas, that CO2, was found right here. Now, if it was true that the radioactivity was already there before we put the sugar water in, then notice after we heat it up, heating is not capable of getting rid of radioactivity. So here's the thing, we're not measuring any radioactivity here, and therefore we have reason to believe that whatever is causing this radioactivity in the headspace must have been coming from what we dribbled on the soil, and the only thing that could be would be the carbon dioxide. So this is consistent with something oxidizing the glucose. Therefore, it's consistent with living organisms on Mars. It's what we expected if there were living organisms on Mars. These are real data. I'm not making this up. This was done in the 1970s, and that is what we got. The NASA scientists were so excited, they said, hey, let's do something. Let's do this. This is going to be fun. Let's scoop that out. Or we can't scoop it out, but let's get another scoop, put it on top of the first one. So they did that, and they scooped it in there. Now, if there were organisms in the soil, what would you predict? Well, if there's organisms in the soil and you repeat the experiment, even on top of the previous one that got heated up, you should get CO2 again. You should get radioactivity in that space. Well, here's what cycle 3 is. There it is right there. And that's what they got. They got, again, radioactivity in the headspace increased. All right? So all of these results are consistent with something oxidizing glucose that was given to the organisms, or if there were organisms, and the thing that probably was oxidizing would be organisms. So this is consistent with there being life on Mars. Okay, what about the other side of the planet? What about Viking 2? Well, Viking 2 did the same thing. Okay, open, scoop, close, purge, dribble the sugar water, and this is what they got right here. Cycle 1, right there. Okay, then what do they do next? Heat up that sample, heat it up, and they get cycle 2. There it is. Okay, now, again, it's basically the same result. Before you heat it, you get radioactivity in the headspace, just like you did the first time on Viking 1. After you heat it, you essentially get no radioactivity. This is all just noise, and that's, again, exactly what they got in Viking Lander 1. So they got excited, and they said, okay, well, that still is consistent with there being life on Mars, so let's take another scoop. And again, they didn't design it so they could scoop the first one out. So let's take another scoop, put it on top of the first one. Now, this time, when they put it on top of the first one, this happened in Viking Lander 1, too. When they put it on top of the first one in Viking Lander 2, it's above the heating coil, right? So what they then do is they check to see if the second scoop is going to release radioactive carbon dioxide when they give it sugar water, and it does. There it is. There's cycle number 3. Now, this time they heat it up again, but remember, the second scoop is above the heating coil. So the heating coil is only going to get the bottom part of that second scoop, not all of it. They're not going to kill, therefore, all the microbes if there are microbes in there. So what would you predict? Well, in the second scoop, you're only killing some of the microbes, not all of them. So do you expect more radioactivity, less radioactivity, the zero radioactivity? Well, we don't expect zero because we didn't kill all the microbes. But we expect less than what we got the first and second times because we killed some of them. And there it is, cycle 4. Notice it's more than the first control where we killed everything, but less than the other two where we didn't kill anything. So again, does this prove that there are microbes on Mars? And the answer is, of course, no, we can't prove anything. But these data are consistent with there being microbes on Mars. So, this is the 1970s. Why are we not running around telling everybody that there's life on Mars? The reason is because the other experiments that they did on Viking all came out negative. They all came out negative. And one of them in particular should have found some sort of organic compound. They should have had some sort of organic compound in that. Because again, these results are indicating organic metabolism, something eating an organic compound. Remember, organic compounds are any compounds that contain carbon besides carbon dioxide and carbon monoxide. So why is it that you eat organic compounds? Why do you eat these carbon-based compounds? Well, because you're made out of carbon. The stuff that you're made out of is carbon. So that means this, that stuff on Mars appears to be eating carbon dioxide, or carbon-based compounds. It appears to be eating glucose. But if it is, then its body should be made out of glucose. It should be made out of carbon-based compounds, not necessarily glucose, but some sort of organic compound. And they didn't find any right there. Now later they did. Subsequent missions, they found organic compounds, but not the Viking experiments didn't. And the other experiments looking for metabolism and looking for other evidence of life came back negative. To this day, we still cannot correctly and definitively interpret this result. We still don't know yet. So that's our goal. We want to try and figure out what's going on. That's why we keep talking about life on Mars. That's why we keep looking for it, because this experiment came back unclear. Now, a lot of people dismissed it early on, and that was probably, in my opinion, a mistake. And they're starting to sort of rediscover it again. This is, we can't just throw this out. And that's our point. This is the going back to the baloney detection kit. All hypotheses have to be followed up, and all the evidence has to be explained. We still can't explain what happened on Mars here. We still don't know what caused this. And until we do, scientists are not going to rest. We're not going to let this go until we know exactly what's going on there. That's just how we think. But at any rate, now, you've seen this experiment, you've studied the experiment. I want you to be able to, again, express the experimental design to other people, talk about how it was designed, talk about what its controls were, what its positive and negative controls were, and talk about what the results were of its controls, and talk about what the results were of its test. Now, here's a little quiz. True or false? This is a control. Okay, this is what was done on Earth, and this is when they put the scoop of soil from California in there. Okay, that's a control. Yes, the answer to that question is true. It's a control. Is it a positive or negative control? Well, it's a positive result. That doesn't strictly mean it's positive or negative. It's a result that we expected to be positive. That's what makes it a positive control. Okay, true or false? This is a control right here. Okay, this is again on Earth in California where they heated up the soil. In this case, this is a control, but it's a negative control because we're expecting to have killed all the microorganisms in the soil sample. And we did. It's quite clear we did. Okay, true or false? This is a control. True or false? This is a positive control. Notice it's a positive result. Okay. This is not a control though. Okay, so this result is not a control because it's the actual test. It's the experimental test. Okay, so here we have a positive control, here's a negative control, and here's the experimental test. Those are the three things. So what's this? Well, this is labeled a control. What is it? Well, it's a control that's done on Mars. It's the negative control done on Mars. So we have two negative controls, one on Earth here, one on Mars here. In fact, a fourth negative control right here. We have a negative control right there. So we have all of these controls that we can run, and this is a test, this is a test, and these three are also tests. This is labeled a control, but it's kind of a semi-control. So why didn't we run a positive control on Mars? Well, think about that for a moment. A positive control means you know there's microbes. The whole point is we don't know if there's microbes on Mars. We're trying to determine if there was. So there's no way to do positive control on Mars safely. We could take microbes from Earth and then use that to test to see if it's working on Mars. The problem is If we do that, then we would contaminate Mars with our own microbes or contaminate our experiment with our own microbes. So that's not really going to work very well. So we can't really do safely a positive control on Mars. But this is the structure. This is what I want you to be able to explain and express. I also want you to remember these results because this is something that we still have to try to work out.